Hey guys, I want to give a quick overview of how DNA, RNA, and protein work in a cell to create the different functions that a cell may do. So we're going to start here with a cell. And so a cell is going to look something like that. And then inside the cell, we're going to have a nucleus. And you might know what's enclosed inside the nucleus is the cell's DNA. And if we zoomed in, this is the DNA is going to look something like this. It's just going to be a long string of it's just a long string of information, really. So it's going to look something like this, where inside that nucleus we have a super long string of all the information the cell ever needs, or any cell in your body will ever need. And so it's going to look something like that. And that's the instructional booklet for everything the cell could ever do. And if we zoomed in here, not all of the DNA is going to be purposeful, good information. There's a lot of information enclosed in the DNA that we'll never ever use. It's just there because it's always been there. But we do have certain segments of the DNA called genes where specific functions in the cell are encoded. For example, there's a gene, might be right here, for maybe your eye color. So a gene right here might be your eye color. There's a gene that could be right here. That's your hair color. There might be a gene right here that tells your liver cells how to function. There might be another gene right here that tells you how much melanin your skin cells are going to produce. There could be another gene right here that helps determine how tall you are. But lots of different genes. Every single aspect of who we are is encoded inside of our genes that encode for proteins that help perform those specific functions. So if we zoom back out and we look at our cell, this DNA, that's a lot of DNA for a little tiny cell up here. And the DNA can't leave the cell because there's so much of it, and there's little tiny pores inside of the nucleus, but those aren't big enough for the DNA to travel through. So what's going to happen is the DNA is going to be copied into RNA, and then the RNA is small enough to leave through the little holes inside the nucleus. So what we're going to do is we're going to make some copies of this DNA. So this DNA might be copied into RNA right here. And then we're going to take that RNA, if it lets me, and we're going to move it outside the nucleus. So for simplicity, I'm going to draw some of these DNA fragments into RNA. And then we're going to take that RNA and we're going to take it to a ribosome where it is coded into a protein or turned into a protein. So the RNA is copied from the DNA. We call that process transcription because the RNA is actually a slightly different kind of language. DNA is going to use the following letters to encode its all of its information. Its entire language is just made up of 4 letters. DNA is made up of A, T, C, and G. And the A's are going to pair with T's, and C's pair with T's, or C's pairs with G's, sorry. So the apple doesn't fall too far from the tree, and you park your car inside of the garage. This might be our way to remember that. And we'll get more into like why this matters in a second. But we have the RNA, on the other hand, has a very slightly different language, but we can very easily convert one language to the other language, because the only difference is that we have A's, U's, C's, and G's. It's the exact same thing, but instead of T, RNA uses U, which stands for uracil. So again, it's the same thing. We have the same rules. A's now pair with U's because they don't have T's. It's just going to be U instead. And then C's still pair with G's. All right. Now, the entire process of what this is going to look like is we have the DNA, again, inside of the nucleus. And then if we want to make a copy of a specific gene to turn into a protein to carry out the function of that gene, we're going to make a copy just like we did here. And then that RNA is going to leave the nucleus and travel to a ribosome. So it's going to travel to a ribosome. So ribosomes, you might know, are the center of where proteins are synthesized. And we're going to take this RNA strand and we're going to send it through the ribosome so that it can be read. The ribosome is going to read it in groups of 3 letters called codons. And every time it reads a codon, it's going to bring an amino acid over. So we might have an amino acid that's brought over for this first segment, and right here, we might have another amino acid brought over for another segment, right here, and another amino acid for another segment, right here, etc., until we have a long strand of a protein. So where everything occurs and what we call everything, we're going to have DNA is going to be converted into RNA, and that process is called transcription. The DNA is in the nucleus and it stays in the nucleus. The transcription occurs in the nucleus, and then the RNA is going to leave the nucleus and travel to a ribosome to create a protein. So RNA to protein, we call that process translation. And that occurs with the help of ribosomes at the location of the ribosome. And then lastly, let's quickly look into how to read the DNA and the RNA to make that protein. So on this gene right here, we might have a DNA segment that looks something like this. I'll do the DNA in purple. So T, A, C, A, G, G, A, C, G, 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 A, and A, T, T. As we said, so we're going to use RNA to copy into a protein. So now we're going to use the RNA. And as we said, the rules are A's pairs with U's, C's pairs with G's. You can pause this video and try to pair, every time you see a T, pair an A. Every time you see an A, pair a U, etc., using these rules right here. So pause the video and try to complete that on your own. All right, so we can fill this in now. U, G, so A, U, G. A is going to pair with U, C, C, U, G, C, CCT, CCU, and then UAA. Awesome. So we got that filled in, and then so that's how we are going to transcribe the DNA into RNA. Lastly, we need to convert the RNA into protein. And to do that, we're going to use a codon chart, which we can find something that looks like this. They look differently, but they all perform the same function. So we'll use that, and then we can look and we can see that when we're going to read this from the inside out. So the first one is AUG. We're always going to use the RNA when we use the codon chart. So the AUG, we have AUG. That's going to code for methionine. So we put methionine down.
To the office, April Chavarria, Sadie Dominguez.
All right, so if we're converting this into a protein, this first one is going to be methionine, MET, right here. The second one is going to be UCC, serine, SER. So these are going to be the amino acids that are going to be forming the protein. So the next one, UGC, cysteine, and then CCU, proline, PRO, and then lastly UAA. UAA is going to be a stop codon, which tells it, hey, we've made the protein, we don't need to keep reading, we're done. So stop. So that's how that all works. The final thing would be that different mutations, which you can read about, but basically if we swap one of these letters, a lot of times that's going to result in a different amino acid, and that can result in a completely different protein. For example, the sickle cell anemia is the results, the difference between your blood cells looking like this and being in a sickle cell shape, like that, is one amino acid difference. There's other more severe mutations. If we add a base pair, if we add one, like we add an A there, then it throws the reading frame off. And now we're still going to read in groups of 3, but we completely messed up the entire rest of the protein. Likewise, if we delete one, then again we're going to read in pairs of 3 and completely mess up the reading frame and end up with a completely different protein. Furthermore, if we mess up the start codon, so the start codon's different. If this is like an A instead, then we'll start at the wrong spot. So we'll keep reading until we get to a start codon. And then finally, if we mess up the stop codon, then we'll keep reading until we find a stop codon, and it'll again mess up the entire protein. So there's different mutations that can happen. Those are different things you can read about.